Hi there, I'm Eirini and I work at the University of Glasgow, and I'm a plant biologist. I'm interested especially in plant photobiology and how plants in general respond to their environment, especially light and temperature. Photobiology is basically the science behind how light is sensed and how it's transmitted within the cell and the organism in order to respond to it. So obviously photobiology is very broad. It's not just in respect to plants. And I was always interested in light signaling, so how light is perceived. So in mammalian cells, there's rhodopsin-based systems, and again, based on lectures, I was fascinated by rhodopsin and how it works. It's a GPCR, which basically is a protein, like a channel, that is a target for a lot of processes, especially biochemical processes for the pharmaceutical industry. But also, it's very important for light sensing and visual perception in mammals. But plants have completely different systems, and that's why I find them extremely interesting, because they have diverse families of proteins in order to detect, so sense light and then transmit this signal in order to change their architecture and development. Can plants see? They can sense light. They don't have eyes, but they have proteins that have eye-like, let's say, domains or structures that can perceive the light and then respond to it. So although they're not the canonical eyes that we have as animals, they actually can sense and respond to light. So in some ways they can see, they can sense the light, and in some ways better than animals, because they can sense light quality, duration, direction, periodicity. So yeah, in some ways they're a little bit more advanced than animal systems. In plants, in particular in Arabidopsis, which is our model plant species, there's 5 different families of photoreceptors. So each one is specialized in perceiving different wavelengths of light. So the first one, based on the electromagnetic spectrum, it's UVR8, so it's the only UVB receptor in nature. So this receptor is a protein that uses its tryptophan amino acids to sense light. And this particular receptor is very important for photoprotection against ultraviolet radiation. So plants can make their own sunscreen to protect themselves against UV, and UVR8 is very important for that. Then there's 3 families of blue light receptors, the phototropins. So these are very important for phototropism, but also other responses, but they got their name based on the original response that they were identified by through genetic screens. So there's 2 phototropins in Arabidopsis. Then there's the cryptochromes. These are highly conserved also in animals, but again in plants they have specialized functions. They're also important for clock entrainment. And there's the Zeitlupe family. There's 3 proteins in that family, and again these are very important for clock entrainment, flowering regulation, and other responses. And last but not least, there's 5 phytochromes. So phytochromes are red, far red light receptors, and these are the ones that actually sense the ratio of red and far red light. So basically, if plants are grown under a canopy, or shade, as we would call it, they can sense this difference in light quality, because the plants that are at the top, they shade the ones that are found on the ground. They basically absorb all the red light, which is very important for photosynthesis. So these plants that are at the bottom, they can actually sense that there's shade, so they try to change their architecture in order to search for light and basically try and photosynthesize. It's very important to know how plants can sense and respond to light, because obviously in nature they're exposed to different light regimes and fluctuating light conditions depending on the environment that they grow. And also we need to keep in mind that light doesn't act in isolation. So when plants grow in a natural environment, they're exposed to different environmental stimuli. So light is one of them. But recently it's been more and more obvious that light, and in particular temperature, go hand in hand. So understanding how plants respond to light to change their architecture in order to optimize growth, and sometimes let's say affect their development, transitions that are irreversible, is very important. And again, because of this connection between light and temperature, it's recently been shown that actually there's common pathways, there's common receptors. So some of the major light receptors, such as phytochrome B, which is a red light receptor, has been shown to also act as a thermosensor. The same for light signaling component that have been shown again to act as thermosensors, or again joint components for light signaling. So basically the plant uses the same pathways or the same proteins to activate genes that lead to very similar architectural responses to a changing environment. So hopefully we can apply some of our knowledge by studying these model species I mentioned, Arabidopsis, into economically important crops, and also try to improve crop quality and also yield, which is very important in response to a changing environment. So I was always interested in biology. I think as a child I was fascinated by the natural environment. I grew up on a small island, so I was exposed to sea creatures, marine animals, but also lots of different plant species. And I always wanted to go abroad to study, in particular biology. I was fascinated. And at the beginning, I wasn't sure exactly what I wanted to follow in terms of biology. I wanted to keep my options open and broad. So I knew I was interested in biochemistry, molecular biology, and then marine biology. And I never thought I would become a plant biologist. It wasn't something I was against, but it wasn't my main passion, let's say, to start with. But it was during my undergraduate degree that I had fantastic lectures, and that sparked my interest in plant biology, in particular plant photobiology. There were specific genetic screens that were mentioned when I was in first year and second year undergraduate at the University of Glasgow. And again, changes of gene expression that were happening in response to light activation, and the photoreceptors themselves. That was an area that I believe at the time it was rapidly evolving, and they were discovering these new proteins and how they work. And once I became interested in photobiology and I attended conferences. Like I mentioned in my talk, Professor Winslow Briggs, I knew that's something that I would love to actually work on, even though I had never worked in a lab before. And once I went to the lab and started working on phototropins, I was hooked. The photobiology community was so welcoming, and attending conferences, it was great for me. And I just loved being part of the community, and I will encourage anyone that is interested in light signaling to go for it. The best part about being a scientist, and especially in academia, is there's a lot of freedom to basically choose the biological question that interests you. Of course, it depends on the funding that is available, but there's always opportunities that you can have little projects, let's say the risky projects that you find extremely interesting. And also the fact that you don't know where your research is going to take you. It depends on, the findings that you will achieve through experiments. On an everyday basis, it's very exciting to design, to have a hypothesis, test it, and find something new, whether it's what you expected or not. And I think that's fascinating. It's basically like having your hobby as your job, and also inspiring the next generation, and looking at people that work in your lab, or students that you teach, how they evolve and they develop. I think it's very rewarding.